Again, viruses are not considered alive. One reason that viruses are not considered alive is they lack the enzymes for making proteins, for making parts that make up that structure of a virus. So they have to rely on a host and its parts in order to make new viruses. So it can't replicate on its own, therefore it's not considered to be alive. So of all the different hosts and viruses that are out there, typically speaking, there's usually a limited host range. So host range being what can the virus infect and target. So some have a broader host range, but still limited. So an example would be West Nile virus. West Nile virus only can target cells in certain organisms, like mosquitoes are affected by the West Nile. People can be infected by West Nile. Birds and horses can also be infected by West Nile. So it's able to infect a few different hosts, but it's still limited. Something like measles is extremely limited because measles only infects people. And how do the cells within these organisms get infected? There's several different entries that the virus can have depending on what type of virus it is. But one type is through fusion, so a lock and key method. So the virus is able to attach to the host cell, the membrane on the outside of the cell. And again, lock and key, it clicks together. And so it would have to be a certain type of cell as well. And what exactly is clicking together? The receptors on the host cell with the capsid, the outside of the viral cell. So whether it's proteins or glycoproteins, or even that viral envelope clicking together with a host cell. So fusion is one way that a virus can get entry. Another type is through injection, which would be with the bacteriophage. A bacteriophage only can infect bacteria, so it has a really limited host range. And when it infects that bacteria cell, it injects its DNA into the cell itself. So almost like a needle going into skin with DNA. Another type of entry method for a virus is through endocytosis, which you might want to look up. But for me, visually, what endocytosis looks like is Pac-Man eating those little yellow dots around, like in an arcade, like on the screen. So the regular cell would be a Pac-Man, or the host cell would be the Pac-Man, and the little yellow dots would represent the virus. So endocytosis, like eating or engulfing the virus. So those are three different ways that viruses make its way into the host cell or organism. Now, as I think I mentioned, viruses can only infect, with like that lock and key, certain types of cells. So something like the cold virus, rhinovirus, it only will infect our upper respiratory cells. So our cells that are within our mouth, our nose, and our throat. Once we or anything is infected by a virus, the virus can quickly reproduce within those cells, and viruses can mutate easily once they are in our cell, which makes it really hard to treat and to have medications for. So RNA viruses, there's no error checking mechanisms for RNA to make the proteins that the virus needs. So because RNA is going to code for these viral proteins to be made, RNA can change. So if the RNA changes, the protein is going to change that the virus needs. It's hard to get medication to target something that often is changing, which isn't necessarily good news for us, but it would be good news for the virus. Here is a simplified viral replication cycle of how a virus is able to replicate and hijack its host cell. So the very first thing, whether it's fusion, injection, or endocytosis, the viral DNA is going to, or RNA, is going to get into that host cell. The DNA is going to get read by the cell because it is a nucleic acid. It is going to read for viral proteins to be made. So when transcription and translation take place and now we have viral proteins in that cell, those viral proteins are going to do damage basically within the host cell. The viral proteins are going to tell the host cell what to do basically and assemble new viruses. There are two methods to which viruses are able to reproduce and affect their hosts. One type is called a lytic cycle. And in a lytic cycle for a virus, this is where the virus is going to use the host machinery or parts in order to replicate, assemble, and release copies of itself, of the virus itself. There's an example called virulent phages. This is where the virus is going to do the same thing. So virulent phage is an example of a lytic cycle. But in the end, the host cell dies through lysis or apoptosis. So apoptosis is programmed cell death, so the cell is going to die that way, or it's going to lyse or burst. So in this case, the viruses are going to be released and the cell is dead with a virulent phage, which again is an example of a lytic cycle. Phages are the best understood of all viruses. So they are, since this diagram is showing you the lytic cycle of a T4 phage or a bacteriophage. Again, a type of virus that targets bacteria cells. When I'm going to be discussing the steps of this lytic cycle, I'm going to start at the top and then work clockwise towards the right, the bottom, and then the left of the screen. So there are 5 main steps, even though it kind of just shows you 4 pictures here. So starting up at the top of the screen, the very first step of this lytic cycle is attachment. So that bacteriophage needs to attach to its targeted cell, and in this case, a bacteria cell. The second step, still up at the top of the screen, the second step is where the bacteriophage injects its DNA into its host cell. Once the host cell has that new viral DNA, the phage DNA is going to circularize, and a lot of the times, again, that's how it's going to be encased within that virus. That happens, and then in still the second step, the host DNA is being broken down or being degraded. The third step, so moving towards the bottom of the screen, so 6 o'clock, at the very bottom, at this point now, the host bacteria DNA is completely degraded, and now the virus has taken over that bacteria cell. And the phage or phage DNA and the proteins for the virus are being synthesized and assembled. The next step, going towards 9 o'clock, is where the phage is assembled itself. So the macrophage, however you want to say it, It's going to be assembled within that bacteria cell. And then step 5 at 9 o'clock still is where the cell is actually going to lyse or break open. And all those bacteriophages are going to be released in looking for another host cell to infect and do this whole lytic cycle all over again. The other type of viral reproduction is known as lysogeny or lysogenic cycle. So in this case, the host cell is not destroyed. In this case, the cell is going to go on and make, the host cell is going to go on and make more and more and more copies of itself. But the difference is the viral DNA is incorporated into that host DNA genome. So every time the cell makes a new cell, it has viral DNA within it. Examples of this, so 2 examples, and of course they have different names. So when you have a lysogenic cycle taking place within a bacteria cell, it's called a prophage. So again, bacteriophage or phage DNA, along with that virus, would be called a prophage. When you have an animal cell that has viral DNA in it, it's called a provirus. So oftentimes what can end up happening, we have a lysogenic cycle taking place, the cell has viral DNA in it, and it can turn into a lytic cycle or vice versa, where then the cell is going to burst, depending on radiation, environmental factors, chemicals. So often one cycle can turn into a different cycle, which I'll show you. There's a pretty good diagram on the next slide. Anytime that a cell is using both cycles, it's called a temperate phage, and it's indicated by the symbol lambda. So again, when you see that, that's going to be like a type of virus that's using both cycles, and the cell is surviving or lysing, vice versa. Hopefully this diagram will better explain the last slide, showing you the lytic cycle versus the lysogenic cycle. So we already saw this diagram on the left of the lytic cycle, where very quickly the bacteriophage is going to attach and then insert its DNA. The phage DNA turns into a circle, and then the host DNA is degraded. Then the phage DNA and proteins are going to be synthesized, assembled. Down the road, the cell is going to lyse, releasing all those bacteriophages or viruses, looking to infect another host. Now, what makes a temperate phage is where after the DNA, the virus DNA, has been injected, and that phage DNA is now a circle. There is a choice, basically. Again, there's no thought going into these, molecules don't think. But depending on the environmental factors and what's going on with the cell and radiation and chemicals, either the cell is going to stay in the lytic cycle, or it's going to switch into the lysogenic cycle, where again, the viral DNA is going to be inserted into that genome, the host genome. And then the cell is going to divide, and have a piece of that viral DNA within its genome. So if you look smack dab in the middle, again, this is going to be kind of part of the lytic cycle turning into the lysogenic cycle. So there's a choice. The environment says, for whatever reason, the environment of the cell says, hey, we're not going